Okay, thank you. Cool. Hello, everyone. My name is Matouš Elphick. I'm an undergraduate student studying computer science at Newcastle University. And today I'll be talking about some work that I helped with during my industrial placement last year at the Francis Crick Institute in the electron microscopy science technology platform. And that work is CLEM-Reg, an automated registration algorithm for correlative light and volume electron microscopy. So let's start by trying to answer the question, what is VCLEM? Correlative light and volume electron microscopy aims to make use of the advantages of both modalities whilst avoiding their drawbacks. So fluorescence microscopy is a light microscopy imaging technique which creates contrast by tagging macromolecules in living cells with fluorescent markers. And there's a couple of advantages, which include being able to do compatible with live imaging, meaning we can capture cell state over time. We can also functionally label our biological sample, meaning we can capture certain things that we're interested in, such as activation state. However, due to the diffraction limit of light, fluorescence microscopy is generally a little bit lower resolution. And we also have less biological context because we can only see the things that we tag. So volume electron microscopy, on the other hand, denotes a set of high-resolution imaging techniques used in biomedical research to study the 3D structure of cells. And due to the fixation process, the cells are generally very dead, meaning we can not do any live imaging. And unlike fluorescence microscopy, electron microscopy is non-specific, meaning it doesn't inherently provide information about specific molecules or compartments in the sample. However, electron microscopy does address some of the limitations associated with fluorescence microscopy, as it's able to achieve orders of magnitude high resolution, whilst revealing the underlying biological context. So we can harness the benefits of both modalities to capture high-resolution images whilst retaining the functional labeling to verify what we are seeing. And due to the different resolutions of both modalities, to harness the benefits of VCLEM, an additional post-processing step of alignment must happen. And there are a number of tools to do this, such as ECCLEM or Big Warp. And these tools generally work by relying on the manual identification of precise spatial regions in both modalities. For example, small subcellular structures or prominent morphological features. And by manually placing a landmark at the identical physical position in each modality, spatial correspondence between the two can be established. And a set of these landmarks can be used to compute the transformation between these image volumes, bringing them into alignment by an iterative optimization process. But the problem, however, with such techniques is that this requires sub-micron alignment between data sets, which is especially difficult when you're dealing with data of different resolutions. And also the determination of a landmark is subjective, meaning we could potentially introduce biases into the registration process. And lastly, the finding of landmarks is quite time-consuming, especially when we're dealing with three-dimensional data. And we also see there's a diminishing return in the alignment by placing more landmarks. So to try to address these issues, we've developed CLEM-Reg. So as input into this workflow, we have our multiplex fluorescence microscopy channel and the volume electron microscopy data. And the first step is to automatically detect landmarks in both modalities. And due to mitochondria generally being well distributed and abundant in normal healthy cells, by segmenting them, we can create, in both modalities, we can create masks, and from these masks we can create landmarks. And then we sample a number of these landmarks from both segmentation masks to create 2 point clouds, one for the fluorescence microscopy and one for the electron microscopy. And then we can leverage point cloud registration techniques such as coherent point drift to align the two together. And we can use the same transformation used for the point clouds and warp the multi-channel fluorescence microscopy to ultimately align it with the volume electron microscopy, resulting in the automatic alignment of the VCLEM dataset. So one of the first steps is to segment the mitochondria in the fluorescence microscopy. And to do this, we employ traditional image processing techniques. And the first step is to scale the data, and this is done using min-max scaling. And this ensures that the thresholding process is consistent across data sets. And the next step is to apply a Laplacian of Gaussian filter. And this essentially enhances the edges and detects features where there's high amounts of intensity change. And lastly, we create the segmentation mask using a dynamic threshold. And when we put this all together, we're able to create segmentation that look like something like this. And as you can see, it's not exactly perfect, but the main thing we're trying to capture in this step is the distribution of the mitochondria in the cell itself. The next step is to segment the mitochondria in the volume electron microscopy. And to do this, we leverage the MitoNet deep learning model, which is based on the panoptic deep lab architecture. Panoptic segmentation essentially combines two types of segmentation, that's semantic and instant segmentation. And for those that don't know, semantic segmentation is essentially delineating pixels in the image into classes, whereas instant segmentation is delineating objects within images. And when applied to our data, the results are incredibly good, and it's able to provide out-of-box level performance for the segmentation of mitochondria that are sufficient for many tasks and good for our use case. Next, we must extract the landmarks from the segmentation masks of the mitochondria from both the electron microscopy and the fluorescence microscopy. And to do this, we randomly sample a number of landmarks around the membrane of the segmentation masks. This results in 2 point clouds, one for the electron microscopy and one for the fluorescence microscopy. And these point clouds are far too dense, however, and the registration between the two would be far too computationally expensive and take far too long. So as such, an additional downsampling step is performed to create a more sparse point cloud for each modality, as you can see. So to align the fluorescence microscopy and the electron microscopy, we use the coherent point drift algorithm, also called CPD. This algorithm iteratively aligns a point cloud to a target point cloud by finding the alignment that minimizes the distance between points. And CPD does not require the same number of points in each point cloud, and generally it's pretty agnostic to noise, so even the point clouds aren't the same, it doesn't really matter. And all that matters in general is that the global structures of the 2 point clouds are similar. So as you can see, the small fish obviously is structurally very similar to the large target fish. And when we applied this to our use case, we see the fluorescence microscopy point cloud in red over a number of iterations improves its alignment to match that of the electron microscopy point cloud in blue. And the main output of this step is the transformation of the original fluorescence microscopy point cloud with the electron microscopy point cloud. And so once the alignment between the 2 point clouds has been determined, we can use this same transformation and apply it to each of the fluorescence microscopy channels. This finally creates a fully aligned VCLEM dataset that looks something like this. And so here we are comparing the results of the alignment of these 4 channels using CLEM-Reg versus doing it manually. And qualitatively, we can see the results are very similar, but we wanted to know how accurate these techniques are quantitatively. So to quantify the alignment, the fluorescence signal of a non-landmark organelle, such as lysosomes, was segmented by thresholding, and then we measured this agreement of this segmentation to the manual segmentation of the same organelle, but in the volume electron microscopy. But the problem, however, with this is due to the fluorescence microscopy being non-isotropic in Z, the fluorescence microscopy segmentation masks would be much larger than that of the same organelle, but in the electron microscopy, meaning traditional segmentation metrics such as intersection over union or the F1 score would not work. And as a quick reminder, ultimately, the aim of CLEM is to be able to assign the fluorescence microscopy, to assign identity to objects in the electron microscopy using the fluorescence microscopy. So as such, the agreement between the fluorescence signal and the manually segmented target structure is assessed by calculating the true positive rate. And in this graphical example, we have the fluorescence signal of an organelle in green and the same organelle in the electron microscopy in gray. And the first thing we do is register the two. So after registration, we calculate the intersection between the manual segmentation of the electron microscopy and the threshold segmentation of the fluorescence microscopy. From this, we can calculate the true positive rate, where the pink represents the intersection between the electron microscopy object and the fluorescence microscopy object. And this is divided by the total area of the pixels of the electron microscopy object. So where the alignment is perfect, we would expect the TPR value to be 1, and that would mean that the whole of the electron microscopy object is essentially encompassed by the fluorescence microscopy object. So if none of the, however, on the flip side, if none of the electron microscopy object is encompassed by any fluorescence microscopy, we'd expect to see a TPR value of 0. So in this data set, where you can check it out on the Empire sort of ID in the top right, we've manually segmented the endosomes in this FibSem data and measured the true positive rate of these 4 manually segmented endosomes against the fluorescence thresholded segmentation. And we did this for both CLEM-Reg and a manual registration algorithm. And from this, we see that in general, the results of CLEM-Reg are pretty good, and we see a mean TPR value across the 4 endosomes of 0.74. But expectingly, we see that by manually selecting landmarks, we do get a better alignment with a mean TPR of 0.83. And in this data set, where you can check it out on the Empire ID at the top right, we've manually segmented 5 lysosomes in the electron microscopy and measured the TPR again between the same lysosomes but in the fluorescence microscopy. And we see that one of the lysosomes aligned using CLEM-Reg was actually better than the same but using the manual technique. But overall, again, generally the manual technique was better with a mean TPR of 0.66, whereas for CLEM-Reg it was 0.48. So to ensure the ease of use of this tool by a range of people with varying computational sort of ability, we created the Napari CLEM-Reg plugin. So this essentially wraps the entire workflow as one sort of widget, and you're able to run the entire workflow with one click of a button, which is pretty cool. And I've also split the workflow into its individual steps so that if you could only want to use sort of parts of the entire workflow, then you're able to do that with your own data. And yep, so then the key takeaways of this talk should be that the automatic registration of VCLEM data is now possible and can even be done with one click of a button. We have made 2 open source VCLEM datasets publicly available, which you can check out at the Empire links I showed earlier. And in general, the workflow in theory can be applied to any two modalities. So provided you're able to create segmentation of the same target structure in both modalities, you can then in principle apply the same sort of latter parts of the workflow to it to create the automatic registration. And if you want to check the preprint for this, you can scan the QR code on the left. And if you're interested more in the code, then you can scan the QR code on the right, which will take you to the GitHub page. And of course, I'd like to thank everyone in the CLEM-Reg team for this, which was a very cool project and a good part of my placement. And of course, thank you, and I'll take any questions.
3 minutes under. Any questions for Matouš? I have 1 or 2.
Hi, great work, thanks. I have 2 questions. The first one is, does CLEM-Reg output affine registrations only, or can it also do nonlinear stuff? And the second question is if you've worked with other organelles as well, apart from mitochondria.
So I'm not sure about whether or not we did it with other mitochondria, or other organelles other than mitochondria, but I don't think so, no. In terms of the first question, I think we do offer both, I'm pretty sure, yeah. But yeah, that's it, really. Yeah, we do have both options available, I think. So you can select, I didn't mention this, but there's multiple registration algorithms that you can select there. So depending on your needs, you can select one that works best for your data.
I was wondering, what's the what technique of light microscopy is it? Is it confocal or wide field?
I think it's confocal. I'm pretty sure. Yeah, I'm pretty sure it's confocal. As you can see, I'm a computer scientist. My speciality isn't the actual acquisition, more the. It's got answers. Yeah, it's the computation, of course.
And I was also wondering, with the true positive way of measuring success, isn't that quite highly likely, considering that the fluorescent microscopy is much bigger than your EM?
Yeah, so I guess, yeah, so the problem with that, because it is just so much bigger, if you were then to take into like other equations, for example, it would be punishing it based on the fact that it's bigger. And ultimately we just care about the organelle being encompassed by some fluorescence microscopy. So we can say definitively this organelle is this type, you know, from the fluorescence microscopy. So I think, yeah, there is a sort of, there wasn't any like properly nice way of quantifying this, but it's sort of like the best thing we could come up with at the time.
So I suppose you could rotate your alignments and see how likely it is just by randomly hitting something. You could then have a baseline, because if you've got a cell absolutely jam-packed full of mitochondria, then your likelihood of hitting mitochondria just by random might be quite high. We
wouldn't measure the mitochondria because that's the target organelle. So that's why we just selected non-landmark organelles, because, like, for example, lysosomes may be a little bit more sparse. We would use that as a metric, but for mitochondria, like you said, it's so abundant. That's why we use it to actually measure. We don't use it for the measurement of or quantify the alignment, rather than just to actually create the alignment in the first place. Yeah, thank you.
One more quick question before we move on.
So I was just wondering if you can comment on why do you think like there is the manual segmentation still, the manual alignment still works better, and what areas doesn't do well with?
Yeah,
and
so
whether increasing the Z resolution of your confocal images is going to make results better.
Yeah, so I think we found that in general, the alignment is generally better where the mitochondria are abundant. So I think, hold on, I'll go back to one of the slides. Some of the lysosomes, for example, were in regions where there weren't many mitochondria available, and we'd expect that basically the registration process is the best essentially around regions of dense mitochondria, whereas because we selected we've selected lysosomes that are not necessarily within this sort of big sort of cloud of mitochondria, the registration isn't going to be as good in that one area. And yeah, in general, like because of the fact that when we segment the fluorescence microscopy, the structure is similar, but there obviously isn't a one-to-one sort of point cloud between the two. So the registration is never going to be as perfect, and that's why we do expect that being able to manually select landmarks where you can sort of spend your time and find the things that look best will garner better results. Yeah, sorry, was there another question?
Whether increasing the Z resolution of your confocal images is going to make the alignment better.
I'm not too sure if that would. I'm not sure, but maybe I have a think about that, and I can maybe talk to you after.
Thank you.
Cool.
Right.
Thank you.